the common between all. So it will give you the journal pathway. For this we can go into this style and we can change it to color. Welcome to another video tutorial. In this video, I will tell you how we can create and interpret the results of RNA-seq data through this Venn diagram. So before I go into the detail and interpretation, let me tell you how we can go to this web tool to create these Venn diagrams. So first of all, you need to open Google and you need to write Venny 2.1. Actually, this is an online web tool. Here is the first link. You can just simply click here and open in the new tab. You can see here this is actually the interface of Venny 2.1, and here you can see we can upload the list of all genes which we have. So let me show you the list of DEGs which I have already. You can see here I have already the list of DEGs. And actually I have three treatments. You can see the first treatment was control, and here I have applied two treatments like treatment 1 and treatment 2. So in this experiment I want to know that what were the common genes between CK and treatment 1 and what were the different genes between CK and treatment 1. And similarly we can also compare that what were the different and common genes between treatment 1 and treatment 2. So at a time we can compare CK with treatment 1, similarly CK with treatment 2, and similarly with treatment 1 with treatment 2. So any of these two combination we can compare with this help of tool. So what we have to do, we just simply go back and here we will write the name of treatments. So for the list 1 we can write here, because we have the control, so we can simply write like CK, and here we have treatment 1, so we can write treatment 1, and here we can write treatment 2. So now we just need to simply copy paste our all genes. So first we can copy the CK genes. Yeah, we just will paste here. You can see it will show you the number of that genes, and similarly we can go at treatment 1 and copy these genes, go back to treatment 1 and paste here. You can see here as we will paste, it will automatically calculate that there were actually the 50 genes in the control and 56 genes were in the treatment 1, and you can see about 40 genes were common between these two. But before we further interpret, let me paste the third one as well. We can see here the treatment number 2. We can copy all these genes and paste here all the genes. Now you can see here it has created the Venn diagram automatically. Here you can see this actually the control has 50, the treatment 1 has 52, and the treatment 2 has 55. So if we try to look the overlapping genes between all these treatment, so you can see there are actually the 35 genes. So you can see here when I have clicked on 35 genes, so it has shown the list of all those 35 genes. If we try to compare control with treatment 1, you can see here it is showing only 5 genes. So when we will click on this 5, so it will show you the list of those 5 genes. So if you want to get those genes which were different between treatment 1 and control, so you will click here. So similarly, if you want to get those genes which were common between treatment 1 and treatment 2, so you will click on these 16, because there are totally these 16 genes. So in fact, if you want to find your major genes or the candidate genes which you think that will play the different role, So then you should know your own story, that what were the difference between these control and treatments. So you can try to find those genes between the common genes, mostly these genes like which were the common between all. So it will give you the general pathway, but if you want to find the candidate genes, you can try to compare between only two treatments, like the difference between treatment 1 and control can be find through this 5. Similarly, the difference between treatment 2 and treatment 1 can be explained by these 16. But if you want to find the common pathway, so then you have to focus on these 35 genes. So now I will go that how we can make it suitable to export that so that we can use it in as such it in our research article. So for this we can go into this style and we can change it to color. So you can see here now this figure has turned into color. So similarly from here we can use different lines and sizes. You can see here we can increase and we can decrease. Similarly, we can also change the font size from here and we can adjust it according to the need. From here, we can adjust and change the fonts. So I will go with the first one. And now if you want to save this figure and want to use it in your research article, so you can click here, left click here, and you can save this image as PNG. So you will just click here. You can see here a high quality figure has been saved. So hopefully this video will be helpful in determining the overall pathways and the candidate gene pathway using this Venn diagram. So if you have still questioning the use of this, please let me know in the comment section. Thank you for watching. Bye bye.